Hi, it's Dr. Phoenix. Today we're going to be looking at mutations. When a lot of us think about mutations, we think about deformed creatures like five-legged sheep, two-headed turtles, eight-legged frogs. And I think a lot of our understanding came from Hollywood, from films such as Teenage Mutant Ninja Turtles and films like that. But in actuality, mutations can be some of our common characteristics that we carry around in the human population. For instance, blue eyes themselves was a mutation many thousands of years ago that has been passed on from generation to generation. So what exactly are mutations? If you look at this sequence of events here with me, we know that DNA is in the nucleus of the cell. DNA contains genes that codes for a particular protein. Those genes have to be transcribed into RNA, and then RNA is translated into a protein. So basically, a mutation is going to be some sort of a change in a DNA molecule that will affect the coding, that will ultimately affect the way the protein is built. So mutations are changes in genetic material. So here we have a strand of DNA, and the complementary strand of messenger RNA, and this is the amino acid sequence that it would code for. Well, a mutation would be a change in that normal DNA sequence. For instance, instead of a T here, an A was mistakenly placed in that spot. Well, that would change the mRNA as a result, which would change this particular codon. UCC no longer codes for the same thing as ACC, and we would see a different amino acid placed in that second spot. Okay, so that is basically what a mutation is. It's some sort of change in the genetic material. Now, how does this occur? Many times mutations occur just through some sort of malfunction during cell division, through either mitosis or meiosis. Other times this can occur through exposure to certain chemical or physical agents, and those things are collectively known as mutagens. A mutagen is something that can lead to a mutation, like radiation or certain harmful chemicals that we could be exposed to. Fortunately, mutations are very rare. They are normally automatically repaired during the replication process. DNA polymerase goes through and repairs those mutations that might happen to take place. So fortunately, they're very, very rare. But when they do occur, when mutations do occur, the cell that receives the mutated gene will pass on that gene to every generation after that. So all of the daughter cells that come from that mutated cell will end up being affected. So what does this mean? If that cell that's mutated happens to be a gamete, which is a sex cell, then when that cell fertilizes the other gamete, the new organism will have this mutated gene in every one of its cells. So the new organism will have that mutated gene throughout its entire body, and it will cause some sort of a genetic deformity or genetic disease, as we see in this group of pictures. Now, if the mutation occurs in a body cell, such as your skin, then that mutation won't be passed on to any future generations. That mutation will be limited to those particular cells in those tissues. So for instance, if a mutation occurs in a skin cell due to exposure to the sun, then that could result in a tumor that forms in the skin, and that could ultimately be removed, but it's not going to be passed on to future generations. Now, mutations can come in a number of forms. They can be small mutations which will only affect a particular gene, a single gene, and those are called gene mutations. Or mutations could produce changes that could affect an entire chromosome, and those are referred to as chromosomal mutations. In this unit, we're just going to look at gene mutations for now. So gene mutations involve a change in just one or a few nucleotides, and they can also be known as point mutations because they just occur at a single point in a DNA sequence. There are three types of point mutations that we're going to look at, or three types of gene mutations, and they're called substitutions, insertions, and deletions. Here's a substitution mutation. This is our normal DNA sequence, the normal mRNA complementary sequence, and the amino acids that are coded for as a result. A substitution would be where one nucleotide is mistakenly placed in the wrong spot. So this is supposed to be a C in the DNA sequence. A T was placed there mistakenly. So that is the nucleotide in the middle of this 3-base sequence. It's going to cause a change in this codon, and this codon will now code for a different amino acid than what it was supposed to code for. So what we're seeing here is a substitution mutation that results in a change in just one amino acid in the entire protein. Now, you might not think that this is very consequential, but I'll show you in a minute that it could be very consequential. Now, the one thing I want you to look at here is that even though a mutation, a substitution mutation, can result in a different codon, remember that sometimes there are multiple codons that can code for the same amino acid. So it is possible that a change or a mutation can code for a different codon but still code for the same amino acid, which would result in no effect. So that is possible. I'd like to show you this sentence that's made up of 3-letter words, and these 3-letter words would represent codons, basically, that would spell out a particular amino acid. So each of these would be amino acids that are coded for by a particular codon. Well, does the meaning change in the sentence by changing one codon or one amino acid? The fly had one red eye. The fly hid one red eye. Of course, the meaning of this sentence does change. So changing one nucleotide that results in only changing one codon, therefore one amino acid, can definitely have a dramatic effect. And the most common example that is usually pointed out in a substitution mutation is this example of sickle cell anemia. Hemoglobin is a protein that is in red blood cells, and it's used to carry oxygen. And there is a mutation in the population that results in a change in hemoglobin. Now, the hemoglobin protein is about 150 amino acids, and with sickle cell hemoglobin, there is a change in just one of those amino acids. It's a result of a substitution mutation. So instead of GAG, we have GTG in the gene. All right, it results in one amino acid being different. And the result of that is, instead of hemoglobin taking on more of a globular shape, it takes on more of a longer shape. And that results in the red blood cells becoming sickle-shaped rather than having this normal donut shape. So the effect is dramatic, even though it is tiny. All right, the other two types of point mutations are known as insertions or deletions. And the effects of insertions or deletions are even more dramatic than substitutions. Because what happens is when you insert or delete a nucleotide into a sequence, that is going to cause a shift of the codons from that point on, from the point of the insertion or the point of the deletion. The codons will shift from that spot on. And so because of that, these are collectively called frame shift mutations. So here's what a frame shift mutation would look like. All right, here we have our normal DNA sequence again with the normal mRNA complementary base sequence showing the codons, and here are the amino acids that are coded for by these codons. Let's say between the A and the C, a T is accidentally placed. Right? All the other nucleotides are still present. So what happens now, instead of TAC being the first group of three, now it's TAT, and everything else gets shifted. And so you can see that the first codon now is not AUG, it's AUA, which codes for a completely different amino acid, and every codon after that is different, coding for a different amino acid. So if we use our little three-word sentences again, if we place a Q right in front of the H, okay, we can see how the three-letter codons are all going to shift. QHA would be the next three-letter codon, which codes for nonsense basically, and every codon after that is going to be different, resulting in a completely different protein being formed. A deletion mutation does something very similar. Here, if we take out the H, for instance, everything's going to get shifted to the left. THG is no longer our first word now, it's TEF. And every codon after that is going to be changed. Again, resulting in a completely different protein being formed. So what are the significance of mutations? Let's wrap this up. Mutations can have little or no effect if, for instance, the changed codon results in the same amino acid being coded for. That would result in no change. It's possible that the mutation could occur way on the end of a particular gene, and that might not cause a great big change in the shape of the protein that's being formed. So it's possible little or no effect could occur. But some mutations can cause dramatic genetic disorders. We saw that a substitution mutation could result in sickle cell anemia. It can change the shape of the protein. One thing I didn't mention is that a substitution mutation could result in a codon that codes for stop. And if that occurs early in the gene, then there will be no protein formed. And a good example of that might be diabetes. Diabetes is where individuals are not producing insulin. Insulin is what helps to keep our blood sugar down in our blood. And so if we have a mutation in our insulin gene that results in a stop codon early, then we will not produce any insulin at all. And then, of course, the frame shift mutations, the insertions and deletions, can result in a completely different protein being formed, which could be a toxic substance that could ultimately destroy the organism or cause other harmful effects. All right. All right, I hope that's helpful to you. Please feel free to send me any questions that you might have through my big campus or bring them to class. And I look forward to seeing you tomorrow. Have a great day.